We continued to work on DNA synthesis, and somebody who started out as a young postdoc, Dan Gibson, came up with a substantial breakthrough. Instead of the hours to days to years, he found out that by putting 3 enzymes together with all the DNA fragments in one pot at 50 degrees centigrade for a little while, it would automatically assemble these pieces. So it went from a year or so down to an hour. And this is a breakthrough for a number of reasons. Most importantly, it allows us now to automate these processes. So having a simple one-step method allows us to go from the digital code in the computer to the analog code of DNA in a robotic fashion. And this is being scaled up substantially. We proved this initially by, in just one step, synthesizing the mouse mitochondrial genome. So I had 2 teams working, one on the chemistry and one on the biology. And it turns out the biology ended up being more difficult than the chemistry. So how do you boot up a synthetic chromosome in a cell? This took us substantial time to work out. And this paper that we published in 2007, I think, is one of the most important for understanding how cells work and what the future of this field brings. So this is where we describe genome transplantation. And simply by changing the genetic code, the chromosome in one cell, swapping it out for another, we converted one species into another. And because this is so important to the theme of what we're doing, I'm going to walk you through this a little bit. And by the way, these are 2 of the scientists that led this. Carol Luchtig was the one with the breakthrough, John Glass, and there was a large team working with them. So we initially did this by isolating the chromosome from a cell called M. mycoides. And chromosomes are enshrouded with proteins, which is why there was confusion for so many years whether proteins or DNA with the genetic material. We simply did what Avery did. We treated the DNA with proteolytic enzymes, removing all the proteins. Because if we're making a synthetic chromosome, we need to know, can naked DNA work on its own, or are there going to be some special proteins needed for transplantation? We added a couple of cassettes to it, one so we could select for it, and another so it would turn cells bright blue if it got activated. And we found a way to get this genome into a recipient cell, a cell M. capricolum, which is about the same distance apart genetically from M. mycoides as we are from mice. So relatively close, on the order of 10% or more different. We inserted the M. mycoides chromosome into the recipient cell. And just as with the phi X, as soon as we put this DNA into the cell, the protein robots started producing mRNA and started producing proteins. Some of the early proteins were the restriction enzymes that Ham Smith discovered in 1970. They recognized the initial chromosome in the cell as foreign DNA and chewed it up. So now we have the body and all the proteins of one species and the genetic software of another. So what happened? In a very short period of time, we had these bright blue cells. When we interrogated the cells, they had only the transplanted genome. But more importantly, when we sequenced the proteins in these cells, there wasn't a single protein or other molecule from the original species. Every protein in the cell came from the new DNA that we inserted into the cell. Life is based on DNA software. We're a DNA software system. You change the DNA software, you change the species. It's a remarkably simple concept, remarkably complex in its execution. Now, we had a problem that some of you may have picked up on or have read about. We were assembling the bacterial chromosome in a eukaryotic cell. So if we were going to take the synthetic genome and do the transplantations, we had to find a way to get the genome out of yeast to transplant it back into a bacteria. And we developed a whole new way just to grow bacterial chromosomes in yeast. as eukaryotic chromosomes. And it was remarkably simple in the end. All we do is add a very small synthetic centromere from a yeast to the bacterial chromosome, and all of a sudden it turns into a eukaryotic chromosome. The centromere is, when you look at pictures of chromosomes, it's that little piece in the middle of the Y, for example. So now we can stably grow bacterial chromosomes in yeast. So we had the situation where we had the M. mycoides chromosome in the eukaryotic cell. We could try isolating it and doing the transplantation. The trouble is, it didn't work. And this little problem took us 2.5 years to solve of why it didn't work. And it turns out when we initially did the transplantations, taking the chromosome out of the M. mycoides cell, that DNA had been methylated. And that's how cells protect their own DNA from interloping species. So we proved this by isolating the 6 methylases and methylating the DNA when we took it out of yeast. If we methylated the DNA, we could then do the transplantation. We proved this ultimately by, in the recipient cell, removing the restriction enzymes. And in that case, we can just put in naked unmethylated DNA because there's nothing to destroy the DNA in the cell. So we're at this point now where we thought we had solved all the problems. We could create these new bacterial strains from the bacterial genomes cloned in yeast. And we had this nice cycle. We could work our way around the circle. We could add a centromere to the bacterial chromosome and turn it into a eukaryotic chromosome. The advantages, for those of you who work with bacteria, most bacteria do not have genetic systems, which is why most scientists don't work with them. As soon as you put that bacterial genome in yeast, we have the complete repertoire of genetic tools in yeast, such as homologous recombination. So we can make rapid changes in the genome, isolate the chromosome, methylate it if necessary, and do a transplantation to create a highly modified cell. So at this stage, we decided to synthesize the M. mycoides genome. With all these new synthesis techniques, Dan Gibson took this on almost single-handedly. It's 1.1 million base pairs. We started with pieces that were 1,000 letters long. We put 10 of those together to make pieces that were 10,000 letters long. We put 10 of those together to make pieces now that are 100,000 letters long. And we had these 11 100,000 base pair pieces. We put them in yeast. Yeast assembled the DNA. We knew how to transplant it out of yeast. We did the transplantation, and it didn't work. So those of you who are software engineers have debugging software to tell them where the problem is in their code. So we had to develop the biological version of debugging software, which was basically substituting natural pieces of DNA for the synthetic ones till we could find out what was wrong. We found out we could have 10 of the 11 synthetic pieces, and the last piece had to be native DNA to get a living cell. We resequenced it and found one letter wrong, and an essential gene made the difference between life and no life. So it was in the DNA gene, which is important for starting the whole process of binding to DNA. We corrected that error. So 1 out of 1.1 million, we corrected that error, and we got the first actual synthetic cell from the genome transplants. One of the ways that we knew this was a synthetic cell, we watermarked the DNA so we could always tell our synthetic species from any naturally occurring one. This was reported in early 2010, and Science published it shortly thereafter. The watermarks, we watermarked the first genome that we did just using the single letter amino acid code. And we were accused of not having much of an imagination. So for this new genome, we went a little bit further, and I added 3 quotations from the literature. But first, we developed a whole new code where we could write the English language, complete with numbers and punctuation in DNA code. It was quite interesting. We sent the paper to Science for review, and one of the reviewers sent back the review written in DNA code. much to the frustration of the science editor who could not decipher it. But the reviewer's DNA code was based on the ASCII code. And with biology, that creates a problem because you can get long stretches without a stop codon. So we developed this new code that puts in very frequent stop codons, because the last thing you want to do is put in a quote from James Joyce and have it turn into a new toxin that kills the cell or kills you. You didn't know poetry could do that, I guess. We built in the names of the 46 scientists that contributed to this, and also there was a message with a URL. So being the first species to have a computer as a parent, we thought it was appropriate it should have its own web address built into the genome. And as people solved this code, they would send an email to the web address written in the genome. Once numerous people solved it, we made this available. And let me just, I know this is hard to read. The three quotes are the first one, and probably the most important one to Ireland, is James Joyce. To live, to err, to fall, to triumph, to recreate life out of life. Somehow that seemed highly appropriate. The second is from Oppenheimer's biography, American Prometheus. See things not as they are, but as they might be. And the third from Richard Feynman. What I cannot build, I cannot understand. Everybody thought that was very cool until a few months after this appeared, we got a letter from James Joyce estate attorney saying, did you seek permission to use this quotation? And in the US at least, there's fair use laws that allow you to code up to a paragraph without seeking permission. So we sort of dismissed that one, and James Joyce was dead. We didn't know how to ask him anyway. And then we started getting an email trail from a Caltech scientist saying we'd misquoted Richard Feynman. But if you look on the internet, this is the quotation that you find everywhere. And we argued back, this is what we found, and this was what was in his biography. And so to prove his point, he sent a picture of Feynman's blackboard with the original quotation. And what it was, what I cannot create, I do not understand. I think it's actually a much better quotation. And we've gone back to correct the DNA code so that Feynman can rest much more peacefully. So we can actually go much further now. And there's an exciting paper that includes, out of Stanford, that includes John Glass from my institute, using our work on the mycoplasma cell to do the first complete mathematical modeling of a cell. And I was going to show you the movie of their mathematical model, but it has so many different components, it would take about 20 minutes to show. But this is coming out in Cell, I think, next week. It is going to be an exciting change. So we can go from the digital code to the genetic code, and now modeling the entire function of the cell in a computer, going the complete digital circle. We're going further now. We're using computer software to design new DNA software to create new cells. And this is an important part of what I'll be talking about on a Saturday evening. Now, in conclusion, I hope it's starting to become clear, if it wasn't already, that all living cells that we know of on this planet are DNA-driven, DNA software-driven biological machines comprised of hundreds to thousands of proteins, and these are protein robots coded for by the DNA. These protein robots carry out these very precise functions and developed by billions of years of evolution. You make a change in the DNA code, it makes a change in the protein code. That can affect whether the protein's functional or not. It can affect whether the cell lives or not. It can affect the rate that the protein folds, how fast it is degraded. This is all programming built into life that controls the three-dimensional structure. So this is dynamic regulation, and explains most of what Darwin described as Darwinian evolution. By making a copy of the DNA software, the cell can self-replicate, making another copy of itself. As Schrödinger described, all these processes require energy. And from all the genomes we've sequenced, there's a range of mechanisms that cells can provide energy. As you know, if we eat sugar, that's one way to do it. And with simple metabolism, it's been well worked out pathways for decades, how we extract the energy contained in the sugar molecule, converting it into cellular energy that can drive all these different processes. In 1996, we sequenced the genome of Methanococcus cerevisiae, the first archaea and the first autotroph. That means it makes everything from life from inorganic substances, and it makes its energy by converting CO2 into methane. It also uses the carbon in CO2 to make all its proteins and lipids. All these processes are coded for in the DNA. We've shown now using synthetic genomes, when you put new DNA software into the cell, the protein robots read it immediately, start producing the proteins coded for, changing the cellular phenotype. And when you change the DNA software, you change the species. This is all consistent with Schrödinger's code script, an organism's astonishing gift of concentrating a stream of order on itself. Schrödinger, citing the second law of thermodynamics, the entropy principle, the natural tendency of things to go over into disorder, described the notion of order based on order. We get the order from the genetic code, we get the order from its interpretation into protein code. We can digitize life and regenerate life from the digital world. And just as the ribosome can convert the analog message in mRNA into a protein robot, it's becoming standard now in the world of science to convert digital code into proteins, viruses, and now cells. Scientists send digital code to each other instead of sending genes or proteins. And there are several companies around the world that make their living by synthesizing genes for scientific labs. It's faster and cheaper to synthesize a gene than it is to clone it or even get it by Federal Express. An example of this digital biological conversion is we've been working on new ways to make vaccines using synthetic DNA. And we progressed this process very efficiently. And now working with BARDA and the US government and Novartis, BARDA sends us an email containing a test pandemic flu sequence. And we have less than 24 hours to convert that digital code into a virus. We now have that process under 12 hours. We get that to Novartis, and they're ready to scale up production for vaccines. Now, we're in the process of building a much smaller, simpler, what I call digital conversion device. So my digital biological converter is going to work somewhat like the telephone, where it takes digital waves and converts it into sound waves so you can hear it. Only we'll have a small box that you could attach to a computer to, in fact, receive a digital wave and convert it into a protein, perhaps a vaccine, or even a cell. So think about in a future flu pandemic, we could digitally distribute a new vaccine in seconds around the world, because this is biology now moving at the speed of light, perhaps even to each individual home. All life is derived currently from other cellular life. But I think it's just a short matter of time before someone discovers the right chemical cocktail of enzymes, ribosomes, lipids, together with the synthetic genome, to create a simple boot-up system to get cells without a prior cellular history. So let's look at the tremendous progress in this last 70 years since Schrödinger's lecture on this campus, and try to imagine things 70 years from now. Jim Watson's still with us. Whether he'll be with us 70 years from now is an open question, but certainly some of you will be here in 70 years. So in the year 2082, what will be happening? Well, we just saw my friend Elon Musk launch a private space flight to go up to the space station. Certainly within 70 years, we will have colonized the moon and Mars. There's several working on trying to do that. So just think, new life forms for food, energy, production, new medicines, perhaps a vaccine could be sent from Earth to Mars at the speed of light, taking somewhere between 4.3 and 21 minutes to get there, depending on the distance between Earth and Mars. The digital and biological worlds are now becoming interchangeable. Perhaps more importantly, and I'll speak to this again on Saturday evening, synthetic life is going to be the tool that allows us to understand the diversity of life on this planet and hopefully enable new industries to produce food, fuel, clean water, and medicine as we add 1 billion people to the planet over the next 12 years and every 12 years following that. As we'll hear in a few minutes, and you heard in the introduction, Schrödinger's life helps to stimulate Jim Watson and Francis Crick over 60 years ago to help kick off this new era of the DNA world with their discovery of the double helix. One can only hope that this new frontier of synthetic life will have a similar impact on the future. Thank you very much.